from FAFU, and my topic is identification of a new antifungal peptide W1 from a marine Bacillus Amyloliquefaciens. Ruizhe is potential in controlling fungal plant disease. And I will illustrate my project from 4 main points. Here is the background. As we all know, mycositis is the most serious plant disease. At present, the chemical fungicides are used widely, but it's obviously that the chemical substance is harmful to the environment and human health. And meanwhile, the more chemicals used, the stronger resistance will plant disease have. In recent years, biological control made great progress. The terrestrial microorganisms has always been the main source of biocides, but with continued exploration to find new active microorganisms become difficult. So we choose the marine microorganisms to the research because of the unique environment and great potential. And this is project progress. The sediment samples that we use were collected from the Arctic Ocean. We use M1NA Jobel 2216E R2A-LB to culture the bacteria that from sediment samples and use the PDA to culture the fungal. We obtained more than 400 bacterial strains. After initial identification, we found that 28 bacterial strains have stable bacteriostatic activity. And in the red arrow is the W0101. Based on the 16S RNA gene sequence, the strain was a Bacillus, and the conclusion were further confirmed by the genome sequence and ANI analysis. Then we try to isolate the crude protein using the ammonium sulfate. We confirm that at 14% is the optimal concentration. Octa-Pu25 systems were used to purify the crude protein. Firstly, the protein sample were loaded onto H-type DAD sulfurose fast flow column. All the peaks were collected. We found in the fraction D2 has the best inhibition effect. Then we use the H-type Q column for the secondary purification. We found that the fraction Q1 has the best effect. From the SDS page and MIC determination, we can confirm that the protein is what we need and named it as W1 in the red arrow. By LC-MS analysis, W1 was identified as a peptide containing 21 AA with a molecular weight of 2.4 kDa, which is completely matched with a partial N-terminal sequence of pre-protein translocase subunit YEGC of W0101. And the W1 was a helicopeptide, as the picture B. The synthesized peptide W1 also showed a significant antifungal activity in the picture C. To evaluate the stability of W1 putting the protein on the difference physiochemical properties, found that the stability of protein is quite good. To investigate the antifungal mechanism of W1, scanning electron microscopy was used to examine the effect of W1. We can find that the cell surface of hyphae was highly rosined and swelled, while untreated hyphae cells were intact and smooth. These results suggest that W1 could destroy the cell walls of hyphae. After that, we do some application. We do application on the farmed rapeseeds to evaluate its effect compared with the chemicals. As for the chemical fungicide, we choose the carbendazim and the dimethiclor. And we found that 5 times dilute fermentation had a significant control effect against sclerotium. At last is the summary. We identified new marine bacillus and the antifungal peptide. And second, through the research, the peptide showed stability and a significant antifungal activity. In this way, it has potential application value. And we had applied a patent and published a paper on the frontiers. That's all. Thank you very much.